My talk is entitled Beaten Black and Blue, Assessing the Role of Melanization in Territorial Encounters of the Eastern Fence Lizard. A lot of the work I do in the realm of animal communication centers around the role of color signals during aggressive interactions between males. Now, I do a lot with melanin-based color, which is often studied in the context of social communication and can co-vary with other physiological, morphological, and behavioral traits, for example, higher aggression. For example, darker, more melanized individuals tend to be more aggressive. And the size and color of melanin-based markings have been linked to things like plasma hormone levels, like testosterone, which can negatively impact immune function and thus make the animals more susceptible to parasites. So oftentimes when studying animal communication, you'll see a suite of traits that co-vary within a signaler. For example, you'll see links between hormone levels, performance capabilities, degree of melanization, aggression, and parasite load. And these types of relationships have been really well studied in the signaler, but we don't have a great understanding of how receivers interpret this type of information and how they alter their behavior in response to these melanic signals. I study lizards in the genus Scoloporus, which are a great system for studying these sorts of relationships between color signals and aggression and parasites because males of most species are very colorful. They're territorial and polygamous, and when intruders enter their territory, they can do little head bobs to gather information or show off their color patches and attack to show aggression. I'll just focus on one species today, the eastern fence lizard. In this species, males have paired blue patches of skin rimmed with black melanic borders on their abdomen, which are displayed during aggressive male-male interactions. There's lots of individual variation in the size of the black borders and blue patches, and previous work has focused on the role of the blue patches during aggressive encounters, but it's unclear what role these black borders play in territorial disputes. When an intruder with large or small black borders enters a male's territory, how will this male respond? So we went into the field and painted males to have large or small black borders and presented them to territory-holding males and looked at how these receivers responded. When the free-ranging males were presented with intruders with small black borders and large black borders, interestingly, we didn't find any differences in information gathering behaviors. They performed just as many head bobs and took just as long to respond to intruders in both cases. We did, however, find differences in levels of aggression. So free-ranging males attacked and bit intruders with small black borders much more than intruders with large black borders. So this black melanic signal is conveying information that's assessed during territorial disputes. But the differences in receiver response were primarily in levels of aggression rather than information gathering behaviors. We found no differences in how much they head bobbed or how long it took them to respond to intruders with different sized black borders. But they did attack intruders with small black borders a lot more than they attacked intruders with large black borders. So it looks like this black color is conveying meaningful information, but the transfer of this information is immediate. Receivers are able to quickly extract information from these signals without needing further assessment with these information gathering behaviors. So how do parasites come into play? Well, these lizards have neck pockets that are often filled with ectoparasites like mites or ticks, and they can be really conspicuous. Some ongoing work in my lab is revolving around the role of ectoparasites in communication. We're examining the role of parasites in shaping a lizard's color signals, performance capabilities, as well as receiver response. Specifically, we're looking at whether or not these ectoparasites are affecting an animal's signaling system, if these ectoparasites affect a lizard's bite force, and how receivers respond to parasites on the signaler. We literally just started our summer fieldwork a few days ago, so I don't have anything to share with you just yet. Thank you, and I'm happy to take questions.